better place. So again, I'm a medical oncologist. I focus in sarcoma and phase 1 clinical trials. So experimental therapeutics, clinical trials, as well as management of rare cancers such as sarcoma is my forte. I got interested in sarcoma because I was very much interested in phase 1 clinical trials. And many, many years ago, when we were doing the phase 1 clinical trials, the most patients who get referred to us were either sarcomas, because after 1 or 2 or 3 lines of treatment, nobody knew what to do. And that's how my interest generated in these tumor types. And these are the tumors where every patient, every case has its own unique story, unique presentation, unique solutions, and unique treatments. So that's how I started working in this field. And this has been an exciting, exciting journey. I don't see myself doing anything else besides taking care of the sarcomas and trying to find out the solutions of the problems we have currently. We can move to the next slide. So when we talk about the sarcoma, and I'm not going to talk anything new which has not been discussed in the last 2 days. Most of my slides are going to be summarizing what we have discussed so far and so on. So when we talk about the sarcoma, we have to understand that there are some uniqueness to sarcomas and there are unique challenges to the sarcoma. And some of these are like tissue origin, which is a mesenchymal cell. They're extremely rare, they are extremely heterogeneous, and their response evaluation is also kind of like a challenging. So these are some unique things compared to other cancers. Next slide. So their biological significance is disproportionate to their clinical frequency. It means clinically they are not that frequent, but their biology is so variable and that decide their clinical course so variable. So that's a unique situation we are dealing with. Now, sarcomas, as you all know, comprise of 1% of all the cancers in adults. And problem is not only that, the problem is that this 1% is divided into more than 100 different subtypes. And these are not only subtypes, these are different diseases which we have to address and entertain when we decide about the treatments. And most of these subtypes, they vary in their clinical presentation, their molecular and genetic makeup. Even 2 patients with leiomyosarcoma, which is exact same subtype, may be different at molecular level. So these are some unique aspects we have to understand. Next slide. So when we talk about the numbers, and when we compare these numbers with other tumor types, this 1% is same as what we see in multiple myeloma, testicular cancer, esophageal cancer, where you have well-defined uniform treatment strategies. And when you compare with the prostate cancer, breast cancer, lung cancer, colon cancer, the number of sarcomas are so rare. And the problem is it's not only so rare, but they are all heterogeneous diseases. So that makes it more challenging. Next slide. These are some of the examples of various subtypes. And again, this is just a glimpse of soft tissue sarcoma classification. But WHO has its own classification, which gets revised every few years, outlining each sarcoma subtypes. Next slide. They comes in different size, shapes, situations. They can happen any part of the body. Next slide. Different tissue origins, different subtypes. Next slide. Again, they can present as a hidden issue which manifests in different ways. There's no one particular clinical pattern that they will be discovered. Next line. So these 100 subtypes varies from very benign format to highly malignant. Each sarcoma subtypes has different biology and different genetics, and their only shared characteristics is their cell origin, which is a mesenchymal cell. Next slide. Now, the parallel word to sarcoma is carcinoma. So carcinomas on the left side, you see breast cancer, lung cancer, prostate cancer, colon cancer. On right side, you see leiomyosarcoma, liposarcoma, and you can put all the histologies of sarcomas. So these are two parallels. Next slide. But as we don't treat prostate cancer, lung cancer, breast cancer with one chemotherapy, the problem is that we sort of has applied the same principle in sarcomas. Like traditionally, we have been treating all sarcomas who need some systemic treatment with the doxorubicin. But again, treating with one drug, prostate cancer, lung cancer, breast cancer is not right. And treating with doxorubicin to all the subtypes of sarcoma is not right as well. So this generalized approach has led to low response rates, emphasizing the need for more specific treatments for different subtypes. And that also comes to a misnomer we have in sarcomas: no chemotherapy works. No chemotherapy works because we have that kind of a mindset, because we are not doing right chemotherapy to the right patient. Right chemotherapy in a wrong patient may not be effective at all, and rather more deleterious. So those are the approaches we have to focus on. Next slide. So current standard of care in sarcomas, it's a multidisciplinary approach. Surgical intervention, whether surgical resection or other surgical interventions, chemotherapies, which are systemic treatments for sarcoma subtypes which are responsive to chemotherapy, radiation therapy. I mean, these are the three standard of therapies we have currently available. Now, all these modalities, they are very effective for certain subtypes and certain situations, but they are limited by their toxicity profiles, limited by tumor cells developing resistance to these treatments, and that limited effectiveness is a limiting factor in getting a good success in these tumor types. Next slide. So when we talk about sarcoma systemic treatments, we focus is not only on the cancer cells, but what is outside the cancer cells, what is in the immune system around that cancer cells, and so on. So these are different areas we have to think about when we talk about systemic treatment for sarcomas. Now, when we talk about that is not the only problem. The problem is when you give a drug, that drug has to go through a pharmacological process in the body before it reaches to the cancer cell. So there's a role of pharmacology, which includes pharmacokinetics and pharmacodynamics, means how the drug is going in your system, how the drug is absorbed, how the drug is transported to the tumor cells. So this is all a complex phenomenon. When we check effectiveness of a new treatment on the cell line in laboratory, we see that particular drug is highly effective in killing all the cancer cells. But when that drug goes into clinical trials, many times we have seen that that drug doesn't succeed in the clinical trial because the drug had a difficult pharmacology, the drug had difficult absorption, difficult drug delivery system, and so on. So that's a complex system we're talking about. Next slide. So when we focus on the cancer and we focus on various targets in the cancer, on left side, you see a whole tumor microenvironment. On right side, you see a tumor cell. Next slide. And we come to the tumor cell, there are different targets outside the tumor cell, inside the tumor cells, inside the nucleus, and everything. And understanding of these targets has revolutionized our approach to treat these patients. And most of this development has happened in last 1 or 2 decades, where we are understanding the cancer at molecular level rather than bombarding every cancer with a chemotherapy. So this good understanding of the molecular biology of the cancer has led to good understanding of developing new drugs, and these drugs are called as targeted therapies. Next slide. So what is targeted therapies? On a normal cancer cells, there are different targets on the cell surface and inside the cell. And these targets are different than the common cells. So if we can identify these targets and utilize a specific therapeutic modality or treatment which hits these targets, that can block further progression, further development, and further multiplication of that cell. So that's what is the true meaning of targeted therapies. And this target can present outside the cancer cells. As we can see, they can present inside the cancer cells. That causes signaling cascades, and that causes cell proliferation, invasion, survival, and protein translation into the cancer cells. Next slide. So when we talk about various targets in sarcomas, these are different targets available in different types of sarcomas. And these are just few examples. Next slide. And based on these targets, we have been able to successfully develop some drugs, which are listed here. These are all unique approaches based on our understanding of the biology of these cancers. Next slide. So one of such an example that how a smart thinking about developing a new drug for sarcomas, where you identify the target, you find out what that target does, and how you can design a drug to hit that target. This is one of the examples. Most people talk about GIST, but GIST has been a landmark for us. But most recent one is this tumor entity called as pigmented villonodular synovitis, which now has a new name called as tenosynovial giant cell tumor. And when we reviewed and we understood the biology of this tumor or this subtype of sarcoma, we figured out there is one specific target or pathway which is active in this tumor type, which is called as CSF1 pathway. And that leads all the problems associated with this tumor. Next slide. With that understanding, a new drug was developed. We use that, and this is an approved drug called as pexidartinib. And you can see what kind of changes it brings to the patient's life. On left side, you see a patient with right knee tumor, and left side you see, on my right side, you see a patient who has responded to the treatment very well. So that's an example of smartly thinking smartly about what is the cause of, what is the molecular target of that particular cancer or particular types of sarcoma, and how we can design the drug. Next slide. Same patient, we see radiological responses from left to right, showing the highly effectiveness of this particularly targeted therapy. Now, these tumors, TGCT, many years ago, many people have tried different treatments, including chemotherapies, including radiation therapies. Yes, when they're limited in one area, surgery is the best option, but sometime when they comes back again and again, surgery may not be the right answer at that point. I've seen people who has undergone amputations because of these tumors, which is now unthinkable with the advent of this new drug we have. Next slide. Same patient with the different fusion images of the PET scan. Next slide. Now, when we try to talk about target, and that's how we identify the targeted therapy. Now, the question is, how do we identify target for various targeted therapies, including therapies which are not primarily approved for sarcoma? In other term, that is also called as off-label use of drugs. Means you may have a drug which is approved for breast cancer, colon cancer, lung cancer, or any other cancer. But if you have a sarcoma patient and that sarcoma patient has that specific target, how you can utilize that drug. So that understanding is very, very important. Next slide. And the question is, how do we find out the targets? And the answer to that is the molecular profiling, which is a new stuff, just coming up in last few years. And molecular profiling involves analyzing the genetic and genomic molecular characteristics of a tumor in that particular patients. And it allows us identifying specific mutations, specific biomarkers, which are the targets for these specific targeted therapies. In sarcomas, molecular profiling has revolutionized our way of understanding these tumors and to find out the treatments for these tumors. So it's very, very important that especially in patients who has advanced sarcomas, we look into their molecular profile, not only at one time point, but subsequent in the disease course as well, to understand if these patients has developed new mutations, new targets, which we can address with the specific therapies. Now, why it's important? The molecular profiling not only help in finding out the targets, but it also helps in subclassifying these tumors many times. And then we find out actionable mutations where we can be having available targeted therapies. And this approach is called as personalized treatment strategies, because we are testing that particular patient's tumor at a molecular level. And that picture is for that one particular patient. Next slide. Again, benefits of molecular profiling in sarcoma. There is clinical implications. Molecular profiling help oncologists to choose the most effective and personalized treatment, such as targeted therapies and immunotherapies. And it has been instrumental in identifying patients eligible for clinical trials as well. One such example of this impact, as we all know, KIT and PDGFR mutations, which we find in patients with GIST tumors. And that has changed the whole story, how successful we are identifying the right treatment for GIST tumors. Now, some of you may know about GIST tumor, which is also called as a gastrointestinal stromal tumor. Many, many years ago, this was put in label with leiomyosarcoma. And this was one of the subtype of leiomyosarcoma, which would not respond to any chemotherapies or so. So only thing we find out was that that subtype has a specific target, which is the KIT mutation. And that has led to development or identifying the right drug for that specific. Now, leiomyosarcoma and GIST, they're totally two different diseases. It has to be addressed really differently. Now, what are the future outlook? Ongoing research in molecular profiling will continue to uncover new biomarkers. These technologies are being improvised and being developed in such a way that these tests will become very cheap, very convenient for us to do at different time levels in the disease course of the patient with the sarcoma. Now, precision medicine using molecular profiling is the future of sarcoma care. And I cannot imagine taking care of sarcoma patients without these additional tools and without these additional technologies we are developing. Next slide. Now, this is one of the new technology which has just come into play, and most of you might be familiar. Now, when patient's tumor progresses, we would like to know molecular profiling at each time points. But for that, we had to do repeated biopsies, which is not always practical and possible to do. So then a new idea came that when the tumor cells divides, some of them dies, and their DNA releases in the blood circulation. And by picking up the simple blood sample, we can extract those circulating tumor DNA and study them for specific mutations and so on. So this is a very unique technology, which is simplifying our approach. In other terms, this is also called as a liquid biopsies, where we are doing a biopsy based on the blood sample. Next slide. Now, how the circulating tumor DNA assay helps us in management of sarcomas. They help in early detection of the recurrence. Let's say you have treated the patient and you're following them repeat scans every 3 to 4 months or 3 to 6 months and so on. But we don't know when these things can come back. So is there any technology which can detect this recurrence early on? And that's where the circulating tumor DNA may help. And I'll show you some of the examples we have utilized for that purpose. Monitoring treatment responses, again, we'll see some of the example where we started the patient on treatments and how you see the dynamics of circulating tumor DNA. Not only that, the circulating tumor DNA assay guide us to personalized treatment strategies for sarcoma patients. Next slide. So this is one of the example of one of the patient. Patient has left breast angiosarcoma. Patient has been on a targeted therapy, stable disease, then all of a sudden, we saw some increase in her circulating tumor DNA level, which you can see at this level. And though her CT scan of chest, abdomen, pelvis, which are like usual imaging studies we follow every 3 months or so on, CT scans were normal. But when we saw this thing, that made us to order some additional testings on her, and we happened to order an MRI of the liver because her previous lesion was in the liver, and that led us to find out a new lesion in the liver, and that led us to change her treatment, and we changed her treatment, and now you see that her levels are kind of like a flatlining. Next slide. Similarly here, the patient with dedifferentiated chondrosarcoma, he was treated and he had a surgery, chemotherapy and everything, and he was just followed with the circulating tumor DNA. We were also doing the standard of treatment, standard of follow-up with his scans and PET scans and so on. But then all of a sudden here we saw some increase in his circulating tumor DNA, which was kind of like a fluctuating, and then we saw persistent increase. And that led us to do some additional imaging studies, and that found recurrence of the tumor and further treatment for this patient. Next slide. Another example, patient with undifferentiated pleomorphic sarcoma. Scans were normal, but we saw circulating tumor DNA going up. Led us to order additional imaging studies, started treatment. Now circulating tumor DNA is going down again with a new treatment. So these are some of the examples where we see early recurrences, early detections of recurrences, treatment responses, and so on. Next slide. Another example for similar patient with a breast angiosarcoma. Next slide. Now, we talked about the targeted therapies and various tools to figure out various targeted therapies to identify the targets by molecular profiling and role of circulating tumor DNA, identifying these targets, as well as monitoring the treatment responses. Now, the next thing comes into major play is immunotherapies in sarcomas. And I think my other colleague already has talked in more depth information, Dr. Wilkie. Just briefly touching immunotherapies in sarcomas. Now, why immunotherapy is important? Immunotherapies don't address the cancer cells directly, but they rather work on the immune system surrounding the cancer cells. And they excite that immune system and make the immune system identifying the bad cells. And they destroy the cancer cells by this indirect mechanism. Now, the good thing is, unlike chemotherapy, their effect on the normal tissue is minimal or almost nothing. The toxicity and side effect profile is different, and toxicity is not as usual chemotherapies and so on. The toxicity and side effect profile is more related to the immune system. Next slide. So what is immunotherapy? This is a very simplified picture of like how immunotherapy works. So this is the T cell, which is immune system cells, and this is the cancer cells. Now, this cancer cells has a ligand called as PD-L1 and some other expression, which are identified or not identified by these T cells. Now, these cancer cells develop these things as a defensive mechanism where they cannot be identified, and they combine with it and causes inhibition of the immune system, so immune cell doesn't work against the cancer cells. So the new cells, the new drugs has been approved to block these targets, and these are called as checkpoint inhibitors. And these new drugs are called as checkpoint inhibitors. Next slide. When you look at the immune cells, this is the T cell that has different targets around it. And these numbers are much more now than when we created these slides. Some of these are agonistic antibodies, and some of these are blocking antibodies. And the overall purpose is to block, to unleash the effect of T cells on the cancer cells. Next slide. Again, showing the same mechanism how the immune cells works, immunotherapy works. This is the tumor. releases the antigens, and these antigens goes to the antigen presenting cells, and they goes to inactive T cells to active T cells. And this is how the whole cascade of immune therapy works. Next slide. Now, checkpoint inhibitor is one of the aspects of immune system. Immune system has different targets, which you can address different therapeutic approaches. So one, we talked about checkpoint inhibitors. Now there's a new things coming up, and which is already established standard of care in many other subtypes of cancers, such as lymphomas and other tumor types. T cell therapy is a new approach, and there are new developments in T cell therapies in sarcomas specifically. Basically, what T cell means is you have a tumor cells which expresses specific proteins on their surface. Next slide. Yeah. Next slide. Yeah. Sorry, go one back. Yeah. So these are the tumor cells which expresses specific antigen which are recognized by the T cells, and these T cells inhibits the tumor cells. Next slide. Again, these are some of the examples where we have found some targets which can be exploited for T cell therapy, such as NYSO, which is a marker for synovial sarcoma, and some other targets. Next slide. Again, this is a T cell therapy example where the simple mechanism is that you have a patient with a sarcoma, you do apheresis, take their T cells out, and do some engineering of these T cells, combining and modifying them. And then these modified or engineered T cells are infused back in the patient's body, where these T cells recognize the cancer cells, and T cell activation happens, which kills these cancer cells. And that's the effectiveness of these T cell therapies. Next slide. Now, lastly, we will like to talk about role of clinical trials in sarcoma. And this is a very broad subject to discuss. So what is clinical trials? Clinical trials are the research studies to test new treatments or procedures in patients. The aim of the clinical trials is evaluate the effectiveness, safety, and side effects. These are all three important aspects of running a clinical trial. These clinical trials are critical for advancing medical knowledge and improving patients' care. Now, whatever the treatments we are enjoying today, not only for cancer or any other diseases, these are results of, like, there were some patients before us, many generations, who participated in these clinical trials. Not only it benefited them, but they helped in advancing the knowledge about these clinical trials as well. These clinical trials are conducted in various phases, and we'll talk about that. Next slide. There are various phases of the clinical trials, and this is just for our common understanding. Phase 1 means first in human, small group, focused on the safety and partly on the efficacy side as well. So we look into all those things, but main focus is safety of the drug in patients. Phase 2 clinical trials, we enroll a larger group of patients depending on what we have learned from the phase 1, and then we evaluate on their efficacy, effectiveness, and also monitor the side effects in a larger number of patients. Phase 3, which is a larger scale testing, and compare the new drug with the standard therapies. And phase 4 is that once the drug is approved, it has been being utilized in patients in general. And then these are post-marketing phases of the trials, where we are trying to get additional information about what this drug is doing in the community, what kind of side effect profile which we may be noticing, which we have not noticed in phase 1, phase 2, or phase 3 clinical trials. So that's the purpose. Next slide. Why clinical trials are vital for sarcomas? Because sarcomas are rare cancers, we have a limited treatment options. So we have to always be looking for treatment options. Novel therapies, including targeted treatments and immunotherapies, need clinical trials for validation. So as a sarcoma patient, we always try to take advantage of these novel therapies available around us. And these provide patient access to cutting-edge treatment before general approval. So whenever there's a right time in the treatment course, we should take advantage and benefit of these clinical trials. And they addresses the variability in sarcoma subtypes through personalized approach. Very important to focus on these tumor types. Next slide. Clinical trials are important in advancement in sarcoma treatment. They improve our understanding of sarcoma biology, leads more precise treatments, and there are many ongoing clinical trials which combines with the circulating tumor DNA-based monitoring, combined with the immunotherapies and various combinations. And clinical trials helps us to identify effective treatment for rare sarcoma subtypes that lacks standard of options. Next paragraph. And what are the benefits of participating in clinical trials? You get access to innovative therapies which are not yet available in the public. You're contributing, you're not only helping yourself, but you're also contributing to the development of future sarcoma patients' treatments. When you're participating in the clinical trial, you're monitored very closely, not only by the doctor who's treating you, but regulatory agencies, FDAs, and so on. So you get extra care and attention when you are participating in clinical trial. And there's a potential for better outcomes compared to the current standard of treatment. And in many situations, we don't have any other standard treatments available. So it's very, very beneficial. Next slide. Future of sarcoma treatment, clinical trials. Again, I think I cannot stress more than what I've done, that these trials are very important for us to understand better these rare tumor types and to develop new treatments. They're not going to help us to address the issue of sarcoma we have today, but also in the future as well. Next slide. In conclusion, future therapies for sarcoma, again, molecular subtype will play an important role along with the histological subtypes. It is important to understand the critical biological events to define proper therapeutic targets. Routine use of molecular biology to guide initial and subsequent treatments. So molecular profiling, not only at the time of diagnosis, but subsequently during the course of the treatment and progression of the disease is very, very important. And again, coordinated translational and research for targeted therapies as well as immunotherapies is very, very crucial. Next slide. That's all I have to say. Again, I didn't go more specific in depth for particular therapies because there are so much exciting things going on. We at NextGen Oncology, we are running many, many clinical trials, and we are a hub of providing a place for rare indication, rare cancer, sarcoma clinical trials, where we can cut short the whole bureaucratic process of availability of these novel therapies to our sarcoma patients. So thank you so much for your participation, and thank you for this opportunity. I'm here to answer any additional questions.
Yeah, thank you so much, Dr. Sankhala. So there is a question. It says, is the ctDNA checking for the same genetic mutations as the original tumor? If so, is it possible that these mutations have changed due to prior treatment, leading to missed new tumors?
Yeah. So those are important things. And again, these technologies are not foolproof. So don't take this as a grain of salt that, yes, we can detect everybody. I have seen several patients where I've been doing circulating tumor DNA, nothing shows up, but the scan shows up the disease. So, and similarly, in the other way around. So these technologies are in development. They're getting better day by day, but they are not foolproof. So, and again, circulating tumor DNA can be assessed for many reasons. One, where you're doing just to see the recurrence or see the effectiveness of the treatment. So if you have a patient with metastatic cancer, you did circulating tumor DNA, they collect the blood sample, they compare with the original tumor DNA from the original biopsy, that the identification process happens. and then you follow that accordingly. Now, again, you have been following that particular circulating tumor DNA, but yes, this is a valid question that tumors develop new DNAs, new mutations all the time. So that's why doing a liquid biopsy where you can find out more precisely that what new mutations has happened, and then maybe at the time when we have a new biopsies are also. So these are all combined, very complex procedures to follow on. There's no single rule about these things, but they're very exciting technologies as well to keep a watch on.
Great, thank you. We have one more, I think. Is there anything new for myxoid round cell liposarcoma?
Yeah. So again, we were talking about 100 different subtypes of sarcomas. Now, liposarcoma is one of them. Again, liposarcoma is 4 or 5 different varieties. Dedifferentiated liposarcoma behave differently. Different treatment works on that. Similarly, myxoid liposarcoma is totally different entities. It metastasizes in different ways. Myxoid liposarcoma tend to love bones and tend to metastasize to the spines than other subtypes of liposarcomas. Now, there are many novel therapies which are in development for myxoid liposarcoma. Some of them we are already using them, and some of them are on horizon because this tumor also expresses NYSO, which is a specific target. So there are some T-cell therapies, so there are some immunotherapies, and some other combination clinical trials going on for myxoid liposarcoma as well.
Okay. All right, we'll do one more because I know we started late. Are there any treatments for PEComa other than, I don't know how to pronounce this,
FYARRO.
Yeah.
Again, this was one example where we find out exact target, and that target led to development of this new drug, FYARRO, which is an mTOR inhibitor. This new mTOR inhibitor has different pharmacology than the other existing mTOR inhibitors. That's why it was found to be more effective. Now, PEComa patients, the disease course is very variable. The disease course can be very horizontal, and all of a sudden they pick up the pace like any other high-grade soft tissue sarcomas. So if patient has already progressed on FYARRO, we should look into other treatment options, and we should look into what is the new molecular profiling shows, what kind of targets it shows. Is this patient a candidate for combining not only the mTOR inhibitor with something else as well? Occasionally, we do use chemotherapies when we have rapid progression of the disease to calm down the disease progression, and then go back on some targeted therapies. So that's a broad answer to this question.
Okay. Thank you so much. We do have another question in the chat. We're going to have to move on, but if you have time, Dr. Sankhala, and are able to answer that, we greatly appreciate it. We're going to move on to the poll and then to our patient panel. Thank you again, and so sorry.
Thank you very much for the opportunity, and thanks to my daughter, who is my technician assistant while dealing with this glitch.
Awesome. Thank you.
Thank you.
Okay, we're going to move on to, we have the poll up, and then Julie will be up next. Thank you.